And this is Miss Savage and your lesson on meiosis. This is a form of cell division that gives rise to gametes or sex cells that have half the number of chromosomes. So through this lesson, you should be able to describe the stages of meiosis and the change in mass of DNA in the cells that divide in this way, and also explain how sexual reproduction results in variation within a species. Your first task today, you'll do now, is to answer the questions on the board, on the screen that you can see. So there are 3 questions there for you, and you should be able to answer these with prior knowledge. So just pause this presentation for a moment and do those 3 questions in your book. How many pairs of chromosomes are in a normal human cell? How many daughter cells are produced after mitotic division, so mitosis? And are the daughter cells of mitosis identical or different to the parent cell? Okay, so when we talk about meiosis, we need to make sure that we understand how many chromosomes there are in a human body cell and why we have 23 pairs, or why we say we have 23 pairs. So we have a total of 46. 23 of those come from our mother and 23 of those come from our father. So each of those pairs carry the same genes. So they're not identical in terms of the characteristics, but they carry the same types of genes on each of those pairs. The only ones that actually carry different genes, where there's a difference in the number of genes, are the sex chromosomes. These are the ones that determine your gender. So if you look at the picture on the screen at the moment, you will see that the last set, so the last pair of chromosomes, one is quite a bit larger than the other. So the longer one is the female chromosome, the X chromosome, and the shorter one is the Y chromosome, and this causes the male gender. So if you are a female, you have 2 X chromosomes, and if you are a male, you have an X and a Y. So all eggs from females, all eggs only carry a single X chromosome. The sperm from the male can either carry 1 X chromosome or 1 Y chromosome. So it's actually the sperm that determine the gender of the offspring, because all eggs are X, but sperm can be X or Y. When these 2 cells fuse, that then results in a cell that has 46 chromosomes, and the 2 sex chromosomes will either be XX or XY. So with meiosis, this is only actually used to produce gametes or sex cells. All of the cells in our body contain 46 chromosomes, or 23 pairs of chromosomes. All of them, I say, apart from your red blood cells, which don't actually have a nucleus. So in order for fertilization to take place, for these gametes to be able to fuse, they need to have just 23 chromosomes as opposed to 23 pairs. So with meiosis, it starts in a very similar way to mitosis, in that DNA replication has to happen before the division occurs, but there has to be 2 divisions to reduce the number of chromosomes by half. So sometimes this way of cell division is called reduction division because it reduces the number of chromosomes. Okay, so with meiosis, instead of having 2 daughter cells that are genetically identical to the parent cell, we end up with 4 daughter cells that are completely unique. So we'll take you through the steps of meiosis, and a diagram is important here to be able to follow. So the next task is to copy the diagram into your book, and then I'm going to go through each of the stages, and then we'll add some notes to this in a moment. So just pause this presentation here for a moment, copy this diagram and the labels and the arrows into your book. Take about half a page, if not more, take a whole page, just to make sure that you can write little notes on there in a moment. But copy this into your book and then restart the presentation when you're done. So the first stage in meiosis is the DNA replication. This happens in exactly the same way as it does in mitosis. Every chromosome replicates itself and then is visible as like a cross shape, and that is 2 copies of the same chromosome. So these are now referred to as chromatids because they are copies of each other, and they're joined together by a little structure called a centromere. So once the chromosomes have replicated themselves, they coil up and condense so that they're easier to move around. We have the spindle fibres again, just like in mitosis, arranging these copied chromosomes, these chromatids, into the centre of the cell. So there's a new word there at the bottom, which is on the diagram, and it's homologous. So homologous means the same. So the 2 sets of chromatids you can see, and in the bottom of the diagram, these are, for example, chromosome 1, because in a normal body cell you would have 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, and so on. So the homologous chromosomes are the same number chromosomes, okay? So they are lined up in the centre of the cell by the spindle fibres, ready for the first division. Once they have been lined up in the centre of the cell, what happens next is the chromosomes, as they're lined up, are able to cross over. And if they do this, they're actually able to swap sections of DNA. And this is quite literally called crossing over. This is really helpful in increasing variation in a population. And as we know, if you increase variation, that means that not all individuals are susceptible to the same things. So increased variation is actually really good for species survival. So once that's actually happened, these are separated. They remain 2 copies of each chromosome, but they're separated into 2 separate cells. So this is the first division, and this is referred to as meiosis 1. So we still have 2 cells after the first division that have 2 copies of each chromosome. So that doesn't work with fusion of gametes, because then you would still have double the amount of chromosomes, double the amount of DNA. We need to still reduce that by half. So the cells at the top have to have those chromatids separated into 2 further cells. So a second division takes place. The spindle fibres pull these chromatids apart by the centromere so that you have 1 copy of each of these chromosomes in 4 separate cells. So each of the cells that arise from meiosis is genetically unique and also genetically different from its parent. So we put that all together, and what you can see is DNA in the very beginning at the top the parent cell has one copy of each of those chromosomes, okay? The DNA then replicates, so the chromosomes appear in an X shape as they are two chromatids joined together. These are then separated into two daughter cells in the first meiotic division, and then those chromatids are separated so that we can then form 4 genetically unique cells, each with half the amount of DNA compared to the parent cell. This is meiotic or meiosis, meiotic division, reduction division. Each of the 4 cells, the 4 daughter cells that result are, they have half the amount of DNA when you compare them to the parent cell. So just add these notes to your diagram that you put into your book before I went through each of those stages. Pause the presentation for a moment while you do that, and then come back here when you're done. Okay, quick knowledge check for you. So 5 questions. You can pause this presentation here while you complete these in your book. So 5 questions for you to answer. Where are chromosomes found? What does DNA stand for? DNA has 2 main roles. What are they? What is a gene and what is an allele? When you're done, press play. Okay, so next task is to compare and contrast mitosis and meiosis. So have all of these into your book. So write these all down in a list in your book and then add mitosis or meiosis. I'm just going to move the camera down slightly so you can see what I've done. Okay, so you can either draw this as a table and tick mitosis or meiosis, or you can write the name next to the statement that describes it. So for each of these, you need to say, is it mitosis or is it meiosis? So pause this presentation while you complete the task and then come back here when you're done. Okay, so this is your written task and you do have 2 options here. So I would like you to do a 6-mark question in your book, and that is to compare and contrast cell division in mitosis and meiosis, and of course you can refer to the notes that you've just made. If you don't feel very confident in completing a 6-mark question, then you can copy and complete the paragraph at the bottom in blue into your book, and the keywords are given there for you. That will also help with the 6 marker to give you some key terms that you should be including in your 6 marker. So pause this presentation for me while you complete this and then come back here when you're done. So just to check, if you've written the 6-mark question, to get all of the marks from the question, you would need to state how the 2 forms of division are different, where they happen in the body, how many daughter cells are made, how many pairs of chromosomes each process results in, and make sure you describe which of the 2 forms of division increases variation and explained why that is. Okay, so if you want to just check through your work and see if you need to add anything in, you can do that now. Just pause the presentation and restart when you're ready. So just a few more bits of information here. So normal body cells are made by mitosis. As you know, that is for growth and repair. It is only gametes, our sex cells, the egg and the sperm, that are formed through the process of meiosis. These are the only cells in our body that only have 23 chromosomes. The reason for that is that when the egg and sperm fuse, they need to have a total of 23 pairs. So thinking about the advantages of sexual reproduction, we know that asexual reproduction is very quick. It can produce a large number of individuals. There's no need to rely on a mate, etc. But one of the difficulties with that is that reproduction through asexual reproduction means that all offspring are identical. And if there is a weakness, then all offspring will have that weakness. With sexual reproduction and the combination of genes, DNA, from two individuals means that every offspring is a little bit different from their parents and from their siblings. So everybody has a slightly different composition of DNA. So they have slightly different characteristics. Now, what that means is that if there is something that one individual is susceptible to, another may not be. Okay, with natural selection, characteristics that are best suited to the environment will increase the chances of survival. And in much the same way with humans, if we're all a little bit different, those best adapted will survive. Okay, so that just means that not all of us are susceptible to the same things. And that means as a species, we're more likely to survive as a whole. So here we can see some cell division happening. So this is in an onion root tip, and these have been stained for viewing under the microscope. Now remember back to when DNA replication takes place. Just after DNA replication, the chromosomes coil up and become very, very condensed. And that means that the stains are seen very, very darkly. So when the DNA coils up to become a chromosome and a chromatid, it's very, very dense, and you can see the structures there within the nucleus. And you can see quite clearly which of those cells are undergoing mitosis. Because this is a root cell, this would be mitotic division, not meiotic division. So you can see really clearly which ones are dividing and which ones are not. Okay, so that is the end of my lesson today. Don't forget, whether you're in school or at home, you do need to upload a photo of your work so that your classroom teacher can see what you've been doing. Okay, take care of yourselves, Year 10. Bye-bye.